Hi everybody, can you guys see and hear me all right? Just let me know in the chat. Well, hi, my name is Samantha H. I am a senior in high school and I will be going through some Q&A with you guys today. Can you guys just verify for me that you can hear me? Just so I know, so that if for whatever reason you can't, that I can fix that. Okay, thank you, Derek, I appreciate it. Okay, so I was told that you guys wanted to start with some Unit 5. Is that correct? Okay, well, first we're just gonna go through, just some housekeeping. These are our normal weekly streams. I know that we're kind of wrapping up as AP exams are starting this week and next week. Have any of you guys taken your AP exams yet? I took AP Lit today. It was weird to do it online, but it's okay. It's okay. It's not too scary, I promise. You guys are all gonna do great on AP Biology on Monday. Okay. So our next AP Bio review is our Cram finale, which will be 5 hours the night before the exam, so Sunday. We are hoping to do the first half be student-led, so hopefully led by myself and another student streamer, and the second half will be led by teachers. This will be a really fun event, pretty crazy. We are expecting hundreds of people there, but we are gonna get you guys prepared to get those 5s 100%. So if you haven't bought your Cram pass for that event yet, I would highly, highly recommend it. And as usual, follow Think Fiveable on Instagram, Twitter. We also have a TikTok. Guys, go follow our TikTok. It's pretty good. I'm not even on TikTok, but I do like Fiveable's TikToks. And also, I'm going to do a quick plug. If you are into podcasts, Fiveable has recently started our own podcast. It's called Homeroom. It's a place where students can just talk about whatever is on their mind. We have a book club. I appear on the podcast a few times. It's a lot of fun to do. It's a lot of fun to listen to as well. So I would highly recommend checking that out as well. Okay, so this stream is entirely up to you guys and what you guys need. We'll start with Unit 5 and 6 because I know you guys didn't get to that last week, and then we can go back and do whatever you guys still have questions about. If you ever have questions throughout the stream, it's easier if you do it in the Ask a Question box, just so I can see it, because sometimes it gets lost in the chat. And I will be happy to answer those. Okay, so let's start with Unit 5. So we have heredity. So just so I know where you guys are at, like, what are the biggest difficulties for you guys within Unit 5? I'll give you a second to answer. Or if you just need a general overview, which we can do as well. Just everything for Unit 5? Okay. So the biggest things with heredity is just the difference between Mendelian and non-Mendelian genetics. And so your Mendelian genetics are your Punnett Squares. This session will be about an hour. If we have to go longer, we can definitely see. But yeah, we're going to try to keep it around an hour. Sorry. So Mendelian genetics are Punnett Squares. So that's where you have your genotypes and your phenotypes. So, your genotypes being your, like, literally what the code is. So the uppercase B, lowercase b, that kind of thing. And the phenotype being the actual trait that it codes for. So my dark hair, my dark eyes, that kind of thing. Because with the Mendelian patterns, we can use the Punnett Squares to track how offspring might look with homozygous, heterozygous parents, dominant and recessive traits. So can anybody explain to me the difference between homozygous and heterozygous? Just a little pop quiz for you guys. Okay, homozygous, same alleles, heterozygous, both dominant and recessive. Very good, very good. Very important to note, right? Because, I mean, from what I've seen when I did, practice FRQs, they're not just going to give you a Punnett square. They're going to say, oh, this one is homozygous and this one is heterozygous. So you have to think to yourself, that's where you got to slow down. You don't want to rush through reading the FRQs because you want to think, okay, homozygous, you want to make sure they're the same. Heterozygous, you want to make sure they're different so that you don't mess up your Punnett square across. If there's a Punnett square on this year's exam, honestly, who knows? Not me, unfortunately. And like I said, just Punnett squares. I don't have a practice Punnett square for you guys. If you guys have any specific questions about monohybrid or dihybrid crosses, you can go ahead and ask them. But I just felt that they were pretty straightforward and that you guys are probably pretty smart and you can do that. But if you have any questions, I'll be happy to answer them. I mean, I think for me, I always thought the non-Mendelian genetics were a little bit trickier because that's where we have our multiple alleles, our sex-linked traits, our codominance, and our incomplete dominance. And I also have an example of a pedigree because that is also quite a possibility that they'll show you a pedigree and ask you to analyze it or something along those lines. So I wanted to put one up there just so that we could review what all of these symbols and lines mean. Okay, so just going back to non-Mendelian genetics. So multiple alleles is just the kind of thing where it's like, oh, it's not just black or white. It can be one of these many different things. And that's when you have like your little I like to think of them as footnotes. Again, I don't really have an example to show you. If that's something you need to go into more depth about, I can do that if you want to ask a question about it. Sex-linked traits, for this one, I always thought about blood types, or I'm sorry, I don't know why I just said that. For sex-linked traits would be color blindness. So, you know, you hear the stereotype of, oh, only boys can be colorblind. Yes and no. It is much more common for men to be colorblind because they only have one X chromosome, and that's where the gene for colorblindness is. So if they happen to get the X chromosome that is affected and then their Y chromosome, they will be colorblind. Whereas for women, they would have to have it on both X chromosomes in order to be afflicted. So I do believe it is possible for women to be colorblind. It's just highly unlikely. And I also think that it would be of a different degree of men, but obviously I'm not a geneticist, so don't hold me to that. And they're not going to ask you about that specifically, but just knowing that and being able to apply that. And then lastly, we have codominance and incomplete dominance. This one is always one that trips me up. I always mix them up, but think of it as codominance. They both show up. So that's when, you know, if you have a white cow and a brown cow, they might have a spotted cow because both the white and the brown is on there. Whereas with incomplete dominance, neither of them are on there. So that's the example with like a red flower and a pink flower would result, or sorry, a red flower and a white flower would result in a pink flower because the red and the white mix, neither of them completely show their dominance. And is everybody good with how that is notated on a Punnett square too? Okay, I'll give you guys just a minute if you know you're not good on that. You want to kind of go over that? Okay. I would have to find an example of that really quick just because I don't have one prepared. While I'm doing that, why don't we look at the pedigree and can we just review what each of the symbols means? So the square versus the circle, and then colored in versus not colored in, and then the Roman numerals on the side. So if you guys can work on that, I'll find an example for you to show you how that might look. Did you guys specifically want the codominance and incomplete dominance for those Punnett squares? Because I can see what I can do about that. Let's see. Oh, here, I'll make this bigger so you guys can see it better. Or at least I'm trying to make it bigger. Okay, or not. That's good too. Okay. Is the chat just really behind because I'm only seeing one response? But yes, square does mean male. Thank you. Yes, circle then does mean female. Do we remember what the colored in versus not colored in means? Yes, so Satvik, you just mix those up. Shaded would mean that you are affected, and oh, I'm sorry, I misread your thing. You were correct. Non-shaded means it's not displayed. Yes. And then lastly, do we know what the Roman numerals on the side means? Yes, generation. Thank you, Allison. So then that way, if you were talking about it in an FRQ, you could say like Roman numeral 2-5 is a female carrier, or I mean, she's afflicted. So it is an afflicted female and such. And I mean, it's possible that they could ask you to predict what kind of trait it is just by looking at the Punnett square. I think I saw that in a practice FRQ, but it was not on my exam. I took the 2019 exam. I took it last year. I didn't even think about it for this one. If anybody wants to take a jab at what kind of trait it would be, you can go ahead and we could try to figure it out together. Let me give you a second. And then I'll go back and pull up an example of codominance and incomplete dominance on the Punnett square. Okay, Allison, why don't you think it is sex-linked? Well, there are some males that are affected, right? Right, so Carrot has the better explanation that both genders seem equally affected, but I would agree that it's not sex-linked. Because you would see a lot more, yes, you would see a lot more men that would have it if it was sex-linked. Otherwise, yeah, I think that's the best way to go. Yeah, so good. I'm not entirely sure how to pull up to show you a Punnett square with codominance and incomplete dominance. Okay, so with incomplete dominance, it'll look the same as a normal Punnett square would with your genotypes. Like, you'll just have like your uppercase letter and your lowercase letter, and you would just have to know that that would be an incomplete dominance thing. I mean, I think a lot of the times, if that's something you have to know, the question will tell you that. Like, they'll say this trait is incomplete dominance. They give you all the tools you need for success. You don't have to memorize like what kinds of traits are that. So I wouldn't worry about that. With codominance, because it is two separate traits that are equally as dominant, they actually show up as different letters. So if you have, again, like your white cow and your brown cow, you would have homozygous dominant, so both the same. So let's say big W, and then homozygous dominant again, but with your brown, so big B, and obviously all four would then be BW, which would then show both because they are both equally as dominant, right? Okay, so I see I have a question. Okay, how to identify if something is sex-linked? So I think you really just kind of got it here in the chat. With this case, because they're equally affected, you can kind of deduce that it could be sex-linked. Like I said, if you saw that it was happening a lot more in males, but females were carriers, they might show that females are carriers and they might not. But for example, if you had, okay, can you see my cursor? I can't really tell. Okay, let's see. Well, for example, so for Roman numeral 2-5, see, easy for me to explain so you can see it. So if that darkened circle was not darkened, but their son, number 3-5, was darkened, you could deduce that the female was a carrier for it. If that makes sense. Please let me know if that answers your question. If not, I can come back to it. Okay, good. Okay. Do we have any more questions about Unit 5 and heredity before I move on? I'll give you guys a second. Okay, saw another question pop up. Oh, we have a few, okay. Can you explain what a carrier is again? Yes, so in cases of sex-linked, because it only affects the X chromosome, females have 2 X chromosomes and males have 1 X chromosome and 1 Y chromosome. So therefore, a female could have an X chromosome that is affected, but because it's only on 1 X chromosome and not on both, it won't show itself. So we call that person a carrier because that person could pass that trait down to their son or even to their daughter if the daughter ended up having 2 X chromosomes with it, but they themselves would not be afflicted with it because it was not on both X chromosomes. Please let me know if that answers your question, Christina. If not, we can come back to it. How are we supposed to know the genotype of the wild type? They will tell you that. And if they don't, then that's on College Board. Because that's just another thing that in the little blurb, they'd be like, this is the wild type, this is the other type. Okay, Christina understands. Allison, let me know if that helps. There's a lot of things that it seems like you should know. In most cases, thank you, Harrison. In most cases, they will tell you everything you need to know so that you can apply the concept to your FRQ in this case. Okay. Are we ready to move on to Unit 6? Okay. Unit 6 it is. Okay, so Unit 6. This one has a lot going on. And let me just tell you, you do not need to know every little enzyme and every little thing. It's especially nice that this year is OpenNote, because I know for me, I would maybe just have a quick list next to me of the steps or something if this is something that's particularly difficult for you, but not a whole textbook with this whole chapter. Just write down the little things that might be tricky for you, because this one just has a lot going on. So, DNA replication. So over here in our little DNA replication diagram, I mean, just looking at it, it's like, I don't know where to start. What's important to know is your leading strand and your lagging strand. So the leading strand, which reads, I don't know if you can really see it, but it's reading the 3 to the, it's reading the, I don't even, sorry, I'm just trying to figure out if that's even the best place to start. Okay, leading strand versus lagging strand. Leading strand is better because it can just read it all in one. You can see the DNA polymerase is just laying on the matching matching amino acid, and it's not having any issues. With the lagging strand, it's reading it backwards. And so for that, it has to use Okazaki fragments. So basically, it reads it in chunks and brings it over in chunks. And that's really the biggest thing to know with leading versus lagging strand. I mean, it's nice to see and understand that topoisomerase is the one that's uncoiling it, and then the helicase is unzipping it, so on and so forth. But like I said, you do not need to know all of these little things. If this diagram is helpful to you, again, maybe this is something you could have next to you during the exam. But especially with this exam this year, it's not going to be, oh, list the steps to DNA replication. No, it's going to be something along the lines of, why didn't this work? What was missing? Or if this is missing, why won't it work? Kind of thing. So I don't know. So we have a question? Okay. Can you explain the 3 and the 5 thing? Yes, I will do my best. So the 3 and the 5 are just the 2 strands. So I don't know if you can see them labeled. I know it's kind of small. But so basically what happens is there's a 3 strand and there's a 5 strand. And if I'm being totally honest with you guys, I forget which way it reads. I think it's 5 to 3. That could be wrong, but this is just another reinforcement that I got a good score on this exam without knowing that. It is 5 to 3. Thank you, Karen. That's not that's not something you absolutely have to know, but thank you. It is 5 to 3. So it reads it 5 to 3. And I don't know if that makes any sense to you, or how to better explain that, but that's the leading strand when it is able to read it 5 to 3 and it doesn't have any issues with replication. But then when it has to read it backwards, 3 to 5, it can't match them as easily as it can on the leading strand, which is why it has to use the Okazaki fragments. And I know that that really just was not a great way to answer your question. I don't know. Let me know if you have like a follow-up question to that, and I can try to do better. But again, it's just understanding the concept as a whole instead of each little step that you need to know. Okay, yeah, Khan Academy has some good videos. Thank you. Yes, Khan Academy is a great resource. Obviously, Fiveable is a great resource. Yeah, so you can definitely, on the things that you're still a little bit stuck on, you can definitely go study on that a little bit more. Okay, so as you know, after DNA replication, the mRNA travels to translation, which is our next diagram. So on the bottom, I think it looks like a big blue like couch or sofa, that's your ribosome, and that long strand is your mRNA. And so your little T curly things in there, that's your tRNA, and it reads the mRNA in groups of 3, finds the codons, and it matches with the anticodons for the opposite strand. And you can see it makes a polypeptide chain, which I think is a really cool callback to, I believe it's unit 1 of like structure and how polypeptide chains become proteins, which we can definitely go over later in the stream as well. But yeah, like that's protein synthesis. Like it is the DNA replication, and then the mRNA that's made then goes into the cytoplasm, finds that ribosome, and it becomes a protein. I don't know, that's crazy to me. Like that is happening right now in all of our bodies. Crazy. I personally think that's pretty straightforward. Let me know if you have questions about that. I can definitely get to that. I also included just some regulatory stuff within here too, because I would not be surprised if this kind of thing showed up on the exam with operons. So with operons, there's this whole coding region of the strand. And there's 2 kinds of operons. There's inducible and there's repressible. If it's an inducible operon, it's always happening and it's always coding until it has to be turned off. So if there's just a lot of it being made, or they just like there's too much of it, it will be kind of turned off. So usually that happens if something binds to it, and therefore I believe it's the DNA polymerase would not be able to read the strand. And the repressible operon is basically just the opposite. Oh, epigenetic changes are just like the fancy word for that, just for the regulation of that, or if the gene needs to be changed in any way. So I included that word in there just so that if you like saw it, you wouldn't be completely blindsided by it. What happens in each of the sites of the ribosome? If I'm being honest, I don't know. I would be really, really surprised if that's something you had to know. If you want, that's something that I can come back to later and I can look that up for you. But I would say that's probably not something that you need to know for the exam this year. But I can definitely come back to that later if we have time. So I'll keep that in there. So with operons and just again, epigenetics, and then repressible operons, it doesn't code until it needs to be coded, and the repressor will be taken off so that it can be, made, transcripted, translated, all that jazz, until it needs to be repressed again. And I think for me, the diagrams just make it look more complicated than the actual process and the actual idea of it is. But that is something, I do believe that concept was on my exam. And they'll give you a specific operon and you'll be really confused for a second, but then it'll tell you what kind of operon it does, what it does, why it does it, when it does it, so that you have all the information you need to know, and you just have to know, okay, like when would it code, when wouldn't it, that kind of thing. That is the basis for Unit 6. If you guys have any more questions about Unit 6, feel free to ask them. And then we can either keep going unit by unit, or we can just kind of do a Q&A free-for-all for any questions you might have about what might be on the exam. I'll give you guys a second. I'm going to sip some water. Okay. Do we need to know about hormones? I mean, specific hormones, no. But like, if you're talking about like cell signaling and how hormones might play into that, sure, that's not a bad thing to review. But they're not going to make you like identify a hormone without giving you a diagram, if that makes sense. What topics and units do you think our exam will look like? You guys, I really don't know. I think because biology is so interconnected, I would not be surprised if they find a way to include a little bit of everything on the exam. I can just kind of talk about the changes to the exam this year and what the FRQs might look like. You have 2 FRQs, one will be 25 minutes and the other will be 15 minutes. And from rumors, I've heard that one of them will have 10 parts and the other one will have 6 parts. And that's crazy, I know. I don't even want to think about that. So basically answer what you know first, go back and think about all the stuff that was kind of tricky to you. What AP Biology, yes, so because it's 10 questions, like they're gonna find a way to test everything. With AP Biology FRQs, just historically, traditionally, they will have a lot of diagrams and charts. So being able to read things like the operons and understand those little things, but they might show you like a signal transduction pathway for your cell signaling. They might show you, I'm not sure. Yes, we can definitely go over cell signaling and transportation. No, you're all good. Yeah, I know, 10 parts. I'm in environmental science this year, so I'm also looking at like a 10-part FRQ, and it is for sure crazy, and you're gonna see it go to H or I or whatever the 10th letter in the alphabet is, and you're gonna take a deep breath, and you're just gonna start with what you know, because you guys got this, and you don't have to answer everything to get a 5. Like, they have said that. They have said that they're giving you a chance to show what you know. So I would be surprised if there wasn't some sort of chart or diagram to look at. But then again, with it being online, anything goes. So I really don't know what topics and units will be central, but there will probably be a little bit of everything. Ilya, I would be happy to answer your questions about environmental science, but we're going to do that a little bit later in the stream, if that's all right, just because I want to make sure we get through all of the material first. Okay, so do we want to go back to Unit 1, or do we want to kind of skip around a little bit? Oh, do I have more questions in here? Okay. Okay, can we answer the FRQ questions out of order? Yes, just make sure you label what you're doing. So if you want to start with D, make sure that you type D parentheses and then your answer, so that they aren't confused as to what part you're answering. But yes, you can completely go out of order, you can skip over stuff, come back to it 100%. How many parts of the question do you think we should answer at least? I am not sure what is going to be considered a 5 this year. For me, this probably is not what you want to hear. I'm just going to aim for just as much as I can get done. So like I said, just either start with the identifies, because those are typically easy points, or just start with like, oh, this is a unit you're really good at, you know a lot about it, just answer that question first. I would just say, try to get through as much as you can. Are all of the parts related? Like, does one part connect and lead to the next? Most likely. I've seen it possible where they're able to kind of loosely connect an entirely different unit, but it's not going to be something completely out of left field. Like, they're not going to be asking you about a specific operon and then all of a sudden be like, oh, what happens here in photosynthesis? Like, no, like they will all be semi-related. Okay, I took a practice FRQ today and it mentioned cleaving proteins in an enzymatic pathway in order to activate them. Is cleaving another word for changing the protein shape? Yes, so cleaving, I assume that they were talking about introns and introns and exons. So when they cut the introns out of the codon sequence, yeah, had to double check that really quick. Yeah, that's called cleaving, and that can change the protein shape and activation if they're removing a coding region or something like that. And that's typically something of, not a mutation per se, but something that could go wrong. I hope that answers that question. But yes, that's how cleaving would change the protein shape, because it would change the way that it codes. Okay. Oh, can you go over some of the different mutations? Yeah, so you have point, substitution, and deletion. Ooh, go me. Okay, so point mutation is just when you have a string of, just your amino acids, and instead of a T, it's an A. So with that, it could be fine. Like it could just have no big deal, because some like codons code for the same like polypeptide. So sometimes it's not even an issue. Sometimes it changes the polypeptide sequence, and that can be possibly good, possibly not that bad, or possibly bad. With deletion and insertion, what that does is it shifts the frame. So because it reads everything in threes, if you have your group of three and then one gets added here, it pushes everything, and it pushes all of your groups. And with that, I think you can imagine how catastrophic that can be when it comes to the polypeptide chain, because it's going to be completely different. The protein is not going to function in the way that it's supposed to. It would just be something else. So that's the same thing. So insertion is just when it inserts one, and deletion is when it just takes one out entirely. That's really just everything with mutations. Yes. Okay. Do we have any more general questions, Unit 6 questions? Or do we want to go over a certain unit? Do I think pedigrees are going to be on the test? It's possible. I have no way of knowing. The other thing to think about too, guys, is that there are likely going to be so many different versions of this test. I think I mentioned earlier, I took the AP Lit exam today, and I don't think I've talked to anybody that had the same prompt I did, and I talked to a lot of people. So they're going to give out a lot of different versions. So your version might have a pedigree on it. I don't know. Somebody else's might. What about chi-squares? I would be really surprised if they gave you a chi-square, especially because they said no math. What they possibly could do is, because experimental design is a part of it, they may give you a completed chi-square and tell you to just kind of look at the results. But I would guess that's the extent of it. So again, if College Board gives you a chi-square, I would say shame on College Board. I would say as long as you understand the whole idea of null hypotheses and how to understand a chi-square, then yeah. Oh, I'm sorry that you hate pedigrees. I hope for your case then you do not get a pedigree on your exam. Yeah, I would say it's not as likely as a lot of other things, just because genetics never feels super big in AP biology, even though it connects like the whole natural selection and ecology with kind of this whole biochemistry stuff, but that's okay. Yeah, I'm right with you, Derek. I like pedigrees. Then again, I really like the whole genetics, and I like the later half of the course with natural selection and ecology. I'm not a huge biochem person. Yeah, no, I'm with you. You'll probably, like I said, you'll probably be tested on a lot of different things. So hopefully you will be able to show off your genetics knowledge in there. But again, I would say it's not super likely you'll get a pedigree. I can't promise you guys anything. I don't work for College Board. I wish I knew what was going to be on these exams. Okay. Okay, do we want to go over a specific unit? Do we want to just kind of keep keep it open just for questions. What are you guys wanting to do? I'll give you guys a second. Cell signaling and transportation, sure. So good old Unit 4. I actually don't even have a slide for this. All I put for Unit 4 was that. Okay, can you be a little more specific within cell signaling of what you're looking for? Just because that's really general, and I just want to know where to start with that. And you guys can always ask questions about different units if there's something you're confused about or not in that unit. Like proteins and pathways, so like signal transduction pathways, that kind of thing. Sure. So basically, okay, this one can get kind of complicated too, and this is something that likely will be on your test. So basically what happens, a ligand is sent out, and a few things can happen from here. There can either be second messengers, which are your CAMPs, and they can kind of like really amplify the signal, hence the amp. They can amplify the signal so that a lot of cells, both in the vicinity but also further away, can understand, oh, this is what we need to be doing. And the end goal can vary from a lot of different things. Like they could give you an example of when you're injured and your blood needs to start clotting, that can be a signal that can go out. If your like cell needs to start dividing, hence the mitosis and meiosis, that can be a signal that goes out. Or if a gene is ready to be replicated, kind of thing. Those are like examples of some end goals. So basically after the second messenger goes out, it is received, and then hopefully the desired effect happens. And that's really just, again, just understanding the process in general is much more important than all the little things. Again, if this is something that's particularly tricky for you, you could either have a list, just a really short list of like, these are all the proteins I always forget, like the cyclic AMP is this, and I always forget that, or I always forget what a ligand is, or I always forget what paracrine signaling is. You can make a little note, but don't rely on having your entire textbook out next to you. Does that answer your question? Hopefully. I don't know. I just want to make sure you guys know that you don't have to know. Okay, good. You don't have to know every little thing. You'll be okay. You'll be okay. Okay, does anybody have any other questions? About FRQs, about testing, about AP biology content? Any questions? Okay, so how are both questions going to have a diagram? Honestly, I don't know. But you answer the question separately. So it is very possible that they both could have a diagram, if that makes sense. So, you know, you have your first question, you have 25 minutes to answer it, and then you submit. And then it brings you to a whole separate second question. So it could very possibly be that way. But because one of them is about experimental design, it's possible they won't both have diagrams. It's possible neither of them will. Again, 2020 is really just unprecedented. It's really unusual. There's just no way to know. Okay, you guys are totally flooding in. Oh my goodness. Okay. Which proteins do you recommend writing out to remember or think will possibly be of use? Honestly, if you would almost never need to name drop something, I would say unless it's Unit 6, like with the replication and stuff. So if you really want to be careful, you can say, okay, helicase unzips the DNA, just in case they ask, like, identify that. But like I said, usually with specific proteins, they'll give you what you need to know in that little stimulus paragraph. So I would say that it's not necessarily like, oh, these are the proteins you need to know, just whichever ones that you always feel a little tricky with, or that you feel you might need to know. What parts of the technology should you know? Like biotech? I'm going to come back to that one. Biotech is kind of a big thing. Do I have to remember each step for the processes of cellular respiration and photosynthesis? You should know generally the process. So you should know step 1 goes to step 2, like photosynthesis, photosystem 2 to this, to photosystem 1 to this, that kind of, like, yes, but you don't have to know every little detail. And we can kind of go over that too. So Chloe asked, experimental design, question mark. Basically, part of what they are testing you this year is, can you understand an experiment or build an experiment? Can you understand those parts? So they'll give you an example of an experiment and they'll say label the independent variable, the dependent variable, the control variable, the hypothesis, that kind of thing. So when I talk about experimental design, it's that whole idea. Ooh, action potential. Okay, I don't have a diagram for this. Are you talking about action potential in the sense of like activation energy in enzymes? Can you, yes, okay. So basically what you need to know is that like an enzyme lowers the activation energy for the substrate. Basically the way that it was explained to me is that if you think about like, this is a little bit more chemistry, but if you think about everything just kind of whizzing around, they have to bump into each other to have that reaction, and they have to have enough force to do that. Whereas with an enzyme, you don't need all of that energy. So it just costs less energy to do so. I don't know if that was your question, if you were just confused about that relationship. If I didn't answer it, clarify it a little bit more and I can do better for you. Would a thorough review of Unit 1 be helpful for this test, or would it be better to move on to the other units? You should study what you don't understand. Unit 1, what they'll do with Unit 1 is they'll slip it in. The biggest thing I would say is the whole fact of how lipids, how they function in the cell membrane, and how proteins with like the polypeptide chain, how that affects protein structure. So that's why Unit 1 is there, to kind of set up those connections of why those things are important. So I would say, don't memorize everything from Unit 1, but make sure you understand those relationships, because that's something that they definitely can't ask about. Okay, now I'm going to the little ask a question box. Sorry, guys. Do you have to know thermodynamics and water potential? I don't know. I do believe that they are technically in the curriculum, so they're not a bad thing to know, but thermodynamics, I would say probably not. I don't think I learned thermodynamics. Water potential, maybe. They won't make you calculate it because, again, they said no math. So that's not something that I would spend a lot of time to worry about, because that might just be one answer or like one question that if you don't know off the top of your head, you just kind of skip over. Okay. Sorry. Okay, when asked to design an experiment on the FRQ, what should we include? Okay, so you should they'll tell you the parts that you need. They will likely do that. If they don't, make sure that you know all the parts of the experiment, which is basically just, you may or may not have a testable question, which becomes a hypothesis. And when you hypothesize, make sure you have a strong hypothesis, because it doesn't have to be right. It just has to be a hypothesis. So don't just be like, oh, when you do this, like this will change. How will it change? Will it increase, decrease, whatever you're testing for? Make sure you have a prediction of how it will change. You likely won't have to explain your hypothesis, but if you do, explain why you did that, but make sure that you're specific in your hypothesis. You should also include your independent and dependent variables, your control variables, and how you're going to go about testing it, so that they can see that you know how to make sure to get good results. So yeah, just any way that you would normally go about a lab. I mean, that's what experimental design is. They want to make sure that you know how to get good results and not like just the results you need, but that you know how to compare it to what it normally is. They will likely list it out, like make sure you include those things. If they don't, that's what you need to include. Okay, you're okay, Carlin. I was scrolling through that too. Will unit 1 be very important on the test? That's totally okay. Okay, what is the difference between a thesis and a hypothesis and the different types of hypotheses? I don't understand the first part of your question. Basically, the only thing with the two different hypotheses, a normal hypothesis, for example, is, I'm just trying to like think of a lab. Basically, if X increases, Y will increase as well as a hypothesis, because you're hypothesizing when we change this, when we change the independent variable, the dependent variable will act because of that, is a hypothesis. A null hypothesis is essentially just that nothing will happen. So you're just hypothesizing not that it'll change oppositely, but just that the independent variable will have no effect on the dependent variable. Like, do we need a thesis and a hypothesis? No, you don't need a thesis separate from the hypothesis. They might ask for a testable question, but I highly doubt that because a hypothesis helps to outline your experiment because it has both the independent and dependent variable, and it kind of shows what you're testing for. But yeah, so a hypothesis should just show those two things. And then a null is just saying that nothing will happen, because in a chi-square, basically, if you have results showing that it did indeed have an impact on the dependent variable, that's when you can reject the null and say, no, like these two things are related to each other. Okay, let's see. I already answered that one. Okay, feedback. Oh, so like feedback loops? Is that what you're asking about? Okay, so negative feedback in general. Okay, so I'll go ahead and talk about both just because it's a good review. So positive feedback mechanisms, basically it's just saying we need more. Best example of that is childbirth. I forget exactly what it is. So see, there's another example if you don't have to know the protein specifically, but like, I don't know, like adrenaline and oxytocin and stuff like that. Once it's out there, the positive feedback is saying we need more of this in order to maintain homeostasis. Negative feedback is kind of the opposite. It is much more regulatory. So for that, it's just kind of like, oh, right now we have a lot, like our blood sugar is really, really high. We need insulin, I think. we need insulin to take care of that. So it releases a lot of insulin to therefore lower the blood sugar. And that's what's important to remember with negative feedback, is that it's trying to lower what just was amplified, if you will, in order to maintain homeostasis. Can you explain the differences between leading strands and lagging strands of replication, like how each replicates and the different enzymes involved? Yes, I can go back to that slide. I know that this can be a really tricky one. Okay, so leading strand. Leading strand reads from 5' to 3'. And if you can kind of see that like yellowish box, the DNA polymerase is able to match up the opposing amino acid to it. And because it's the leading strand, it reads it in the direction it's meant to, everything's good. With lagging strand, it has to read it backwards, 3' to 5'. And because it's doing that, it doesn't function in the same way that it's meant to in the leading strand. So it uses Okazaki fragments, where it essentially just reads a few at a time and brings it up. So that's why you can kind of see that little part is like literally fragmented, because it's bringing it up to match up with those. So it does it like a handful at a time. For the different enzymes involved, I can just do a quick overview. Again, this is something you likely do not have to have memorized as every single one. But if you really want to, you can like have a cheat sheet next to you or something like that. basically you have your topoisomerase that unwinds your DNA because we know that it's normally coiled, so that it's straight. The helicase unzips it. These are binding proteins. You really don't need to know a lot about those. Your DNA primase essentially just reads it, primes it, so that the RNA primer can match it up. RNA primer, in this case, does it in Okazaki fragments. You have your DNA ligase, probably just connects them. Honestly, not entirely sure. You don't really need to know all of this. DNA polymerase, yep. Yeah, and then DNA polymerase. That's for replication. Those are the biggest things to understand, I guess. Okay, you joined late. But if College Board said no math, does that mean we shouldn't have to conduct a chi-square? Yes, so Taylor, what I said earlier is I would be very, very surprised if they put a chi-square on there. If they really, really want to test chi-square, it's possible they could give you a completed chi-square and ask you to analyze it. But I personally, if College Board put a chi-square on the exam, I would help you guys write it because, yeah, I think that would be ridiculous. So my prediction is no chi-square. Yes, you're welcome. Okay, what other questions do we have? Okay. Okay. Sorry, you may have already done this, but can you explain the difference between describe and explain? Okay, so I have not gone into depth with this. The difference between describe and explain. Okay, I don't know if this will make sense in anybody else's brain, but for me, I kind of do each action verb by point amount, which is not always true. So this is just how my mind thinks of it. This is not actually like the truth of how it's graded. But identify is typically 1 point. Describe, I feel like, is usually 2, and explain is usually 3. In all cases, you should always use complete sentences and you should be thorough. But with describe, if it's like, oh, describe how this happens, you can usually do it within a few sentences. Just explain like how it happens. With explain, typically I think I go more in depth. And I think both of those action verbs make a little more sense in the context that you'll find them. I think that all of the questions for FRQs, you can kind of gauge how in depth you need to go within the context. So I personally would not worry too much between the difference between describe and explain. You'll probably know when you've said enough. And you'll be fine. Guys, we're gonna all take deep breaths. This exam, you guys are gonna rock it. I already know it. Okay, regulation of gene expression, sure. Okay, so that's regulation of gene expression is our operons. It's technically like our feedback mechanisms, and it's a little bit of the cell signaling pathway too. So this is a little bit how it's all interconnected. So it can be regulated through operons. So if it's not something that's needed all the time, it'll have a repressible operon so that it's only made when it's needed. And vice versa, if it does need to be needed all the time, it'll be inducible, it'll automatically be made until it needs to be repressed. Kind of with feedback again, if more of it needs to be made right now, they'll make sure that it's being made through those operons, and similar to cell signaling as well. I know that was kind of a really quick overview, so let me know if I need to go more in depth with that again. Okay, what types of graphs should we look at? Do you mean to study, Chloe? Is that kind of what you're asking? Like, what kinds of graphs should you study? I mean, I don't know. Like, I don't know that I can think of like a specific graph that you would need to study. I think if you're concerned about reading graphs for studying, you can just do practice FRQs with graphs and look at those examples. I don't know. I know that didn't really answer your question, in which case I'm sorry. Yeah, I just don't really know how to answer that. Cyclins and the CDK thing. Okay, you're gonna give me a second. I might have to look my notes up for that one, because that personally is a little more complicated, in my opinion. Oh, I said I was going to go back to biotechnology and then I never did. Is that right? Okay, I'll make sure to do that. It's in my brain. We're going to do that next. Okay, so cyclins, like your cyclic AMP is the second messenger. Sorry guys. I mean, I don't know, like it's not, I don't know, I wouldn't worry too much in the like your kinases. Basically, those are just examples of second messengers. And that's what's important to know, that, you know, goes reception, transduction, response. After your ligands, you got your second messengers, which are your cyclic AMP and or your kinases, which may result in a phosphorylation cascade, which basically is just a whole bunch of things happening at once to make sure you get to your end goal. I'm really sorry if that was not the answer you wanted. I don't know, we have a lot of great study guides here at Fiveable for really specific stuff like that. And if there's stuff that you still have questions about, we are almost always available to ask questions for too, for like that really, really specific stuff. Okay, I know I said I'd go back to biotechnology, so I just wanted to do that really quick before I forget. Okay, with biotechnology, basically with biotechnology is how humans manipulate how like DNA replication occurs to make things that we need. So theoretically, this live stream ends in about 4 minutes, but I'm willing to go a little bit longer if we still have some questions. Okay, so biotechnology, biggest example of biotech is insulin. So, you know, if you have diabetes, it's because your body struggles to naturally regulate or produce insulin. So they obviously manually inject it. But where does that insulin come from? Basically, prokaryotes and bacteria have, well, actually, I'm pretty sure all cells have this, but it's plasmids, so basically it's DNA in a ring. And what we have started doing is we can cut those and replace some of that DNA with a different DNA sequence, so that when it's replicating itself, it replicates exactly what we need, which is insulin. And that's how we create insulin for humans, is by using other cells and using their ability to replicate genes. I forget who originally asked the biotech question. I'm sorry I got back to it so late. I hope that answers your question. If you guys have any more questions, I'll stick around for a little bit, but if not, then we can wrap things up. I'll give you guys a second. Oh, you're welcome. Make sure to come to the Cram finale on Sunday, you guys. It'll be lots of fun. 5 whole hours of AP biology. What could be better? Isn't that so fun? Great, I'm happy to hear it. Hopefully I'll see you there. Okay. Do you have any other questions? Are we feeling good about the exam? Can everybody give me a thumbs up in the chat if you guys are feeling good? There likely will not be another like Q&A. We might do something along the lines of a Q&A in the Cram finale, although I'm not sure because that's supposed to have hundreds of people. But like I said, if you have specific questions, we're always here to help on Fiveable, so you can always reach out. I was asked about predictions on topics for FRQs earlier. I would not be surprised if they managed to do a little bit of everything. Maybe mainly focusing on operons, that's usually a big one. So that's possible. Cell signaling could be possible. Photosynthesis and cellular respiration can be possible. If we have more questions, where can we ask you / anyone? Okay, so if you are ever on the Fiveable site, sometimes a little thing pops up for intercom. If you leave me a message on intercom, I might be able to answer you. Usually I'm on intercom and I can just have people assign it to me. So if you have any specific questions about AP biology, and that actually goes for all subjects, you can ask questions about that there as well. Yeah, so there you go, Chloe. We are always here to help. We want to make sure that you guys are feeling great about the exams. You guys are going to do great. Okay, what are some ways I recommend studying to make sure you get through everything on Sunday for the Cram finale? So start with what scares you, which I mean, I know is not what you want to hear. But so, for example, for me, I was always really bad at photosynthesis and cellular respiration. That was always really tricky for me. So whenever I would study, I know, what if everything scares you? Hey, you're gonna do great. Guys, do not even worry about it. Deep breaths. Fiveable has great resources. If everything scares you, what you should do is you should do a practice FRQ, and you should see what really tripped you up, and start there. So yeah, start with what you're not sure of. Practice FRQs are great. They help you like actually through the process. Think in the way that you are going to be asked for. What keywords on the FRQ should we look for? I mean, you have your classic identify, describe, explain, just so that you know kind of what you'll be doing. Always use full sentences though. That's like, don't just use a word for identify, guys. Like, use a full sentence. I would say other than like keywords on FRQ, make sure you're thoroughly reading it. Don't make a stupid mistake. Where can you find practices? College Board, I believe, released either 1 or 2 practice FRQs that are going to mirror what they're going to look like this year. So like, I believe they have like a 10-part and a 6-part. So I believe you can find that on the College Board website. Other than that, you can do FRQs from past years. They won't be the exact same format, but it'll be the same language and the same concepts to test. So that's not a bad idea either. If you're an AP classroom, there's probably a lot of FRQs in there too, or you could always ask your teacher if they have any lying around. Does that answer everything? Does anybody have any last minute questions before I go? Okay, you are welcome. I am here to help. You guys are going to do amazing on your exam. Where can we find the 2 FRQs? I don't have a link. I know that they did it as part of their YouTube thing when they were doing YouTube videos. I'm not entirely, I think if you Google it, you will probably find it. If you have trouble finding it, Chloe, see if you can find me on Fiveable and I can help you out. But yeah, okay, I'm not seeing any more questions. Okay, is there a way that we can contact you if we have a specific question? Yes, so like I said, if you're on the Fiveable website and you see the little bubble pop up for intercom, it'll look like it's coming from Amanda, but you can do a reply, and it can even find its way to me. You can ask for me by name and I can try to get to you. But yes, you can always reach out on there first. I am usually on there and I am happy to help. Yeah, but you guys are going to do great. Yes, thank you, Harrison. Harrison did a much better explanation of how you can find us on intercom. Okay, well, thank you guys so much. I hope this helped. Yeah, guys, you do not even worry about it. You guys are going to be amazing on Monday. And I'm going to see you all at the 5-hour cram session on Sunday. It's going to be lots of fun. We're going to go over everything. We're going to answer all of your questions, and you're just going to feel so prepared. And as always, think Fiveable. Thank you, you guys. See ya.